Hi, I'm Dr. Leslie Blankenship-Williams, and in this lecture we are going to dive into experimental design as it applies to human physiology. Now hopefully you've learned a little bit about biological research in a general biology class and such considerations such as designing a testable hypothesis, independent and dependent variable, etc. We are going to revisit some of those concepts, explore them in more detail, but definitely through the lens of human physiology. Now, most of the research that's done in human physiology this day and age really looks at diseases. So when we think about diseases that affect humans, we can kind of think about it in two different ways. One branch of research might be looking at how that disease manifests itself. So that's pathophysiology, the physiology of a disease. In other words, so you have type 2 diabetes. What does that actually mean? What does it look like? What's happening inside the body? And then the other branch is concerned with treatments. How do we treat that particular disease? And when I say the word disease, unless you've been living under a rock these last 2 years, there's been a disease that has completely upended life as we know it, and that is COVID-19. So let's actually use COVID-19 as our example to work through the principles of experimental design. Now I'm going to look at it from the treatment side of it. There's been a lot of research that definitively shows the effectiveness of masks and vaccinations at reducing COVID transmission and the severity of COVID illness if you happen to get COVID. But once you do get COVID, vaccinated or not, masked or not, there hasn't been nearly as much information put out about treatments. What do you do? When you go to a hospital, what are they going to give you? And so I would say that the messaging on the treatments has been much more mixed. And part of that is because early in the pandemic, healthcare teams kind of took a kitchen sink approach, meaning we're throwing anything at this disease in hopes of saving patients. We have no idea if it's working. We're just throwing everything at it and hope that something works. And when we think about treatments like drugs that might potentially treat COVID infections, once again, we kind of have two different branches. You can develop new drugs that will treat COVID, but that process takes a long time. So Merck right now is doing that. There's been a number of other pharmaceutical companies that are trying to basically generate new drugs to specifically treat COVID. And the problem with that route is that it takes a long time. Or you can try taking drugs that are already on the market for other purposes and try reapplying them to treat COVID and see if it works. We saw that in 2020 with anti-malarials, and we see that this year with ivermectin. So ivermectin has been a pretty controversial treatment plan that is already approved for human use as a dewormer. So it basically eradicates any parasitic infection that's caused by worms, usually in your GI tract. And interestingly enough, I did not know this before putting this lecture together. The reason why ivermectin was even brought up, I mean, what a random thing, a dewormer. Worms are totally different than viruses, so you would not think that there would be crossover there. But the reason why ivermectin actually was even brought up in the first place was because there was some evidence that ivermectin worked in a petri dish. In other words, if you culture human cells in a petri dish, infect them with COVID, and then use ivermectin, it seems to inhibit viral replication. Okay, so the first thing we want to think about before getting into this experimental design is separating in vitro versus in vivo. In vitro is going to be any experiment that's done like in a test tube or a petri dish. It's an artificial environment outside of the human body. And just because something works in a test tube does not also mean that it's going to work in a body. So once you show that something might have promise in a test tube or in a petri dish, your next step is to use a mammalian model like mice or rats and try the same kinds of experiments in vivo. And I will tell you that 90% plus or more of drugs that look like they work in vitro never make it to humans, either because they don't work in a mammalian body. You are not a petri dish. You're much more complicated. and or it's not safe. Something about it is toxic, or it doesn't have good delivery to the organs that it needs to get to. So in other words, just because something works in a petri dish doesn't mean that it works in you. Another nuance that's important about ivermectin is that the dose, the amount that is required to inhibit viral replication in a petri dish is greater than what is approved for human use. So in other words, you have to use a lot more of it, which we are not allowed to do without going through the same types of long clinical trials that Merck will have to go through with their antiviral pill. So let's assume for a moment that I want to test, does ivermectin work? I don't want to take the media's word for it. I'm a scientist. I want to do this myself. I am going to pretend that I have access to lots of money to conduct this research and lots of resources. In the real world, money and resources and time and feasibility are always constrictions on your experimental design. But to start with, we're just going to assume that we have access to all of those things. And I want to develop a hypothesis. Now, what do I want to test? I want to test if ivermectin works, right? That's my hypothesis. That's not a testable hypothesis. Ivermectin treats COVID infections. Too vague, too amorphous. What experiment are you going to design with that? Whatever you come up with will probably be quite different than what I come up with. This is how we tend to think. We think kind of general. It treats, it works, it's better, but it's not testable. So we need to refine this. And the first thing we need to do is get rid of the word treats and replace it with something that we can actually measure. What does that mean? Well, there's a bunch of different ways that we could assess if ivermectin actually did, treat COVID-19 infections. I could look at ICU stays, death rates. I could look at viral loads. In other words, I could swab up the nose or in the back of the throat and actually figure out how much virus is on that swab. And that's the one that I'm going to pick. So I'm going to refine this a little bit and say ivermectin is going to reduce the viral loads in COVID-19 infections. Now notice I'm making a prediction, but I'm totally fine with being wrong. Meaning I'm not married to this. I'm not saying, yes, this is going to be my hill that I'm going to plant my flag on, and if it's not true, I'll cherry pick until I make it true. I'm simply making a prediction. Ivermectin reduces viral loads in COVID-19 infections. I could have easily made the prediction, ivermectin does not reduce viral loads in COVID-19 infections. It doesn't matter which one you pick. My testable hypothesis still needs more work. As it's stated, it has more detail, but it needs even more. So I'm going to say that I want to reduce viral loads within 7 days. And I'm also going to put a qualifier in there. I'm going to specifically deal with patients. So I'm in humans. That tells me that I'm working with humans. I'm not working with another animal model like chimpanzees. And that because I have patients, that means that they've clearly sought some sort of medical care. So they're not at home, they didn't take a self-test and say, yes, it's COVID positive. They're clearly seeking some sort of care. But this still isn't good enough. I need much more information to get a really solid handle on what type of experiment I might design. So I'm going to flesh out, no pun intended, my hypothesis to this. Patients exhibiting non-life-threatening COVID-19 infections. So that right there tells me that I'm already qualifying my COVID-19 infections to be mild, so they can't be life-threatening. I'm saying that they are going to be taking a single dose of ivermectin, and that dose needs to already be approved for human use within 2 days of symptom onset. And by that, I mean that the symptoms have just started. Real quick, it's generally accepted that antivirals or anything that will have antiviral activity works best early in the course of disease. When somebody's in late stages of their disease, antivirals don't seem to show a lot of efficacy. So it would make sense then that if we want the most efficacy from a drug that might work, we don't know if ivermectin works, that we would try to pick people that are pretty early in their disease. And one way that we can measure for that is days since symptom onset. So a symptom would be a headache, diarrhea, a fever, a cough, sore throat, etc. And now we're going to make our prediction. They're going to experience a significantly greater viral reduction. Significance implies statistics. The word significant means that there is a statistical test that supports the word significance. I'm not going to get into statistics in my class, but that's what that word means. It does actually, in a literal sense, have significance. You don't just throw it out there. It means there's a statistical test that supports your use of that term. So significantly greater viral reduction 7 days later compared to similar patients who take a placebo. So notice that I've already added in another group of people who are not going to be getting ivermectin, but are going to be getting a fake pill of some sort. So we'll talk about the placebo in a little bit. All right, look at how my hypothesis has evolved from thought, it works, it's going to be used to treat it, to nuance, where now you have basically the bones of an experiment. You have a pretty good idea about what your experiment might look like. And whatever experiment you design from that testable hypothesis will be pretty similar to the experiment that I design, because we've removed a lot of the vague and amorphous terms. So whenever you're developing a testable hypothesis, here are some pointers. First and foremost, it has to be testable. In other words, you need to have a conceptual and feasible experiment that could potentially test whether or not your hypothesis is supported or not. So for instance, what we just described is testable. COVID-19 sex is not testable. God exists is not testable. Whenever possible, you want to have your measurable outcome explicitly stated. You don't want to use amorphous terms or vague terms or ambiguous terms. You want to explicitly state what are you measuring, and if at all possible, it should be quantitative, not qualitative. And you want to remove all subjectivity so that the way you interpret the results would be the same as anybody else's interpretation. So that means words like better, ivermectin works better than monoclonal antibodies. What does better mean? Ivermectin works. What does that mean? It treats it. What does that even mean? Your concept of a drug working might be totally different than my concept of a drug working. You might think a drug works if it cures me within 5 days, and I might think a drug works if it reduces the death rate by 10%. So that's too ambiguous. We've got to really dial it in and make sure that our terminology is precise and nuanced, and preferably quantitative, not qualitative. And then go ahead and make a prediction. You can say, we're going to see if ivermectin has significantly lower viral loads, or you can just say ivermectin will have significantly lower viral loads. You don't need to be omniscient. You cannot see the future. I mean, if you could, you'd be God. So you are trying to make a prediction, and then you're going to run a test to see if the data supports your prediction. And it's okay if it doesn't. A lot of people have an innate fear of being wrong. Like, oh my gosh, the data didn't support my hypothesis. Human error, human error. And it's like, why do you think there's human error? Could it just be that your hypothesis wasn't true? I mean, I'm not testing you, literally, on your ability to predict the future. The whole point of science is to actually see if the data supports what you've said. And it's okay if it doesn't. So we'll come back to that at the very end. But go ahead and just make the prediction. All right, now that we've looked at some considerations for a testable hypothesis, let's look at some other factors associated with experimental design. I've left at my testable hypothesis so that we can refer to it as we try to tease out these various parameters. So every experiment should have at least one independent variable and one dependent variable. Hopefully you've heard of those terms before. Another way to think about the independent variable is, what are you actually varying between groups that you're interested in? Like, what are you testing? Are we testing 7 days? Are we testing whether or not people have COVID? It's like, yeah, we're testing for all those things, but that's not what I'm really interested in. What I'm interested in is ivermectin. Is a single dose of ivermectin going to reduce viral load? So the variable that I'm testing is whether or not people get ivermectin. Did they get ivermectin or did they get the placebo? So the independent variable is usually the treatment or the thing that is being varied between groups that you're interested in. The dependent variable can also be described as the thing that you're measuring. What outcome are you measuring? The measurable outcome. So what are we measuring here? Well, we're going to measure viral load. So we are measuring viral load 7 days after treatment. We're not measuring 7 days, we're not measuring death rates, we're not measuring ICU admissions, we're not measuring their age, we are measuring their viral load. That's the thing that we're really interested in. So in my class, I have test questions where I give you a testable hypothesis, and I expect you to be able to pull out the independent and the dependent variable. So get practiced with it. Whenever you develop an experiment, if possible, and it's not always possible, you want to include a control group. So when we think about groups of people getting different treatments, some of them we're really interested in, like I'm really interested in the ivermectin group. What's going to happen? Are their viral loads going to decrease? Versus the placebo group, the group that's going to be getting the fake pill. So the experimental groups are always the groups that are getting the treatment of interest. And you will always have experimental groups. So here, our experimental group is going to be the one that is getting ivermectin. The control group is necessary in this experiment because it provides a baseline that you can reference in order to compare your experimental group. So it is not always possible to have control groups. I'll give you an example of an experiment that is not possible to have a control group. You are measuring lung capacity in male versus females. Males tend to have broader rib cages, and so therefore we expect them to have more lung capacity. They can move more air. You have two experimental groups, males and females. Your independent variable is biological sex. Your dependent variable is lung capacity. It's what you're measuring. There is no control group. So control groups are not always possible. But in drugs, when you're looking at pharmaceutical drugs, you always want control groups if possible. It is necessary because it provides a baseline. Let us assume for a moment that we do not have a control group. We simply are getting, 100 patients. They have COVID, they're non-life threatening, they've all come in within 2 to 3 days of symptom onset, and we give them all ivermectin. And guess what? 7 days later, their viral loads are lower. Was it because of the ivermectin or was it because of something else? We don't know. We cannot definitively tie any improvement in viral load to ivermectin without something to compare it against. We need a group of people that are as similar as possible to our experimental group that did not get ivermectin, so that we can look at what their viral loads are doing. So, control groups, always include them if possible. They give you a reference point from which to compare the results of your experimental group. So speaking of things that can affect the outcome, we need to also always be cognizant of other types of factors or variables that can affect the results. And you want to eliminate or normalize or control for these factors as much as possible. So let me ask you this. Are there other things besides ivermectin that might affect the viral load of a person 7 days later? And I'm thinking, yeah, there's a whole lot, right? This is not even an exhaustive list, but I have here a whole bunch of things I could think of. If you have a comorbidity, your viral load is naturally going to be higher. If you're older, your viral load is probably going to be higher. If you're unvaccinated, your viral load is going to be higher. If you're boosted, your viral load is going to be lower. Did you receive some type of other supportive care, other drugs that might have lowered your viral load? What was your immune state? Are you pregnant? That's immunocompromised. Are you receiving chemotherapy for cancer? That's immunocompromised. They're going to have very different outcomes than somebody who is immunocompetent, somebody whose immune system is in tip-top shape. What if somebody came in with life-threatening COVID? That would be different than somebody with a mild case of COVID. What about the strain of COVID? Omicron versus Delta. That absolutely could have an impact. What about how the viral load was measured? So let me give you an example. There's recent research to suggest, and it is suggestive, that Omicron levels are higher in the back of the throat than in the nasal cavity. Typically, nasal swabs are used to test for COVID. And then to assess viral load, we do something called PCR. You probably have heard of a PCR test. And the number of times a gene is amplified gives you some indication for how much virus was on that swab. Well, if Omicron likes to replicate in the back of the throat and not up here, and you're swabbing up here for some of your patients, but there for other of your patients, that's something that can affect the result that is not controlled for. So you're going to need to ensure that whatever procedure you use to assess viral load is consistent or controlled across all participants, whether or not they're in the control group or the experimental group. Now, controlled factors, and that's the phrase I use, controlled factors, and I don't use controlled variables, although your professor might. Controlled factors and the control group are not the same thing. A control group is a group of participants that provides a baseline, a reference point to look at my other data against. A controlled factor is something that I'm keeping consistent across all participants, whether or not they are in the control group or the experimental group. So in my hypothetical experiment, I might say something like, all patients must be between the ages of 20 and 60. They all have to be admitted to the same hospital. They all have to be immunocompetent, no cancer patients, no pregnancies. They all have to have mild COVID. They all have to have an Omicron strain. They all have to be unvaccinated, etc. So these are all things that I'm saying, I can see how these things might affect viral load 7 days later, and I want to make sure that I'm eliminating any of those variables. Otherwise, when I get my results, I don't know if any significant difference is in fact due to my treatment of ivermectin or due to the fact that everybody that was in my ivermectin group was also between the ages of 20 and 25, and everybody that was in my control group was over 80. You see the problem. Okay. So let's talk about something else that we need to consider, and that is replications. So replications are the number of data sets that you're going to get. In our case, one replicant or one replicate would be one person in the control group and one person in the experimental group. That would represent one data set. The more replicates you have, the more confidence you'll have in your conclusions. In other words, if you see a difference and you've got 10,000 patients, you'll feel more confident in saying, yeah, there's truly a difference. I think this difference is real. Versus chance, which can happen if there's a difference, but you have a low number of replicants, like 3. And I'll give you an example. I have a coin. It's a penny. Now, I flipped my coin, and, oops, can't see it there, but it was actually heads. Is it a trick penny? Does it only have heads? I'm going to flip it again. Let's say I flip it again, and guess what? It shows up as heads. Now I've done 2 flips. Heads every time. And I flip it a 3rd time. Heads. So my conclusion is that this is a trick penny and this only has heads. There are no tails on this penny. How confident am I in making that conclusion? Would I bet my life savings on that? No. because I don't have a lot of confidence. That may be what my data shows, but with only 3 replicates, I could have easily gotten all the same heads with 3 flips, right? I mean, obviously that's totally possible. Flip a coin 3 times, you can get heads 3 times or tails 3 times. That doesn't mean you have a trick coin. But what if I were to flip the same coin 100 times and it was heads every single time? And you watched, and I did good flips and real flips. And then I said, this is a trick coin, there's only heads. You'd probably have more confidence in that conclusion because you have more replicates to support that. In the same way, the more replications that you have, the more confidence that you should have in your conclusions that you're drawing from that data. So whenever possible, and again, we're looking at humans and financial constraints, so it's not always possible, but whenever possible, you want more replicates because it gives you more confidence in your results. The experiment that I just outlined was actually done. Not quite the same experiment, but a similar one. And so the title of the experiment or the paper is here. It was published in a prestigious journal called The Lancet. It was actually published in February of 2021. So this is before Omicron and really even before Delta. This is when the virus was circulating in 2020. And again, people had showed that ivermectin was working in a petri dish, so maybe it would work in people. And so this hospital wanted to actually test it out. The link to the full paper is down below, and I encourage you to read it because it shows you what real scientific papers look like. And when somebody says, I did research, that doesn't mean that they Googled and came up with the first 3 things that basically matched their Google search. Real research is real experiments. And then they publish that research in scientific journals. And usually it involves being reviewed by other scientists that are eminent in their field. So that's what real research is. So when somebody says, I did research, and I say, really, what was your experimental design? And they're like, oh, I Googled it. That's not research, that's reading. You read. And your source may have been a media source, or it could be an actual scientific paper. Obviously, I'm a scientist. I'm going to give a lot more credibility to scientific papers than a media soundbite. That's not research. Okay, so feel free to read it. I encourage you to do it. We're going to take a look at this experiment and kind of see what they did and how some of their experimental design corresponds with what we looked at earlier. So you can see here that we have the effect of early treatment. So remember how I said viral infections generally have the best outcome when treated early versus late with antivirals. With ivermectin on viral loads, so that's what we're measuring. They go farther and they actually measure symptoms, so this is self-reported symptoms, and they measure antibody levels, that's the humoral response, in patients with non-severe. So they're looking at, again, not life-threatening illnesses. This is a pilot study, usually means it's going to be kind of a trial, not necessarily incredibly robust. It's just kind of a, let's see if there's anything here, and it usually doesn't involve a huge number of replicates or a very refined experiment. It is double-blind and placebo. Now I'm going to talk about those terms before moving on because we need to talk about what does it mean to be a placebo, a blinded study, or a double-blinded study. So a placebo is going to be any treatment that mimics the real thing, but the patient does not know. So in our case, a placebo would be a pill that looks just like ivermectin, but does not have the active ingredient in it. Placebos are ideal for control groups. Why? Because we're a stubborn species that wants to believe. We want to believe that things are going to work. If we did not have a placebo, or the placebo was clearly distinguishable, and I give a patient something that I say, I think this is going to work, and they believe it's going to work, and they get ivermectin, and they feel better, they might kind of exaggerate how much better they're feeling because they believe they should feel better. Likewise, if I tell a patient, this is a placebo, it's not the real deal, it's not going to make you better, and they don't feel that much better, they might almost exaggerate the other way, going, yeah, I don't feel any better. It's a placebo. I know it's not going to work. So the power of belief ends up contributing to bias. And we want to remove bias as much as possible. So a placebo is used in conjunction with what's called a blinded study. In a blinded study, the patient does not know whether they are getting the real treatment or the placebo. So they are blind to what they are receiving. They're usually informed that they'll be in one group or another, but they're not going to know. And it's necessary because as patients, especially if we're doing some sort of like self-reported, I feel better, I'm sleeping better, I can smell better, that you already have a preconceived notion about whether or not something should work. So blinded studies in conjunction with placebos reduce or eliminate the bias from individual participants. But it's not the only source of bias. Like I said, humans are stubborn creatures. Whoever is administering the study should also not know whether a patient is getting the real deal or the placebo. Here's why. Imagine if I'm a physician and I am watching all of this horrible death around me, people dying from COVID, and I really, really want ivermectin to work. I mean, I really, really want it to work because we need something. So think a year ago before vaccinations were available and people were just like, I don't know what to do for these people. I'm out of options. I want to give these people hope. I want to treat them. I want them to live. What can I do? And someone says, well, in a petri dish, ivermectin showed that it worked. And you're like, yes, okay, let me give it. Do you think I'm compromised now? You bet. So my own wishes are clouding my judgment to be objective. So as a consequence, we want to make sure that whoever is administering the drugs and or collecting the data does not know. They're simply told this is patient A132. Somebody else knows if A132 is getting the placebo or the real deal. You're just handing a pill and being like, let me watch you swallow it. I have no idea what's in the pill. So I don't know. You as the patient don't know. And it removes my own bias as somebody who wants a treatment to work, and your bias as somebody who wants a treatment to work. So whenever possible, studies should be double-blinded. Again, being blinded means that the patient does not know whether they're getting the real deal or the placebo. And being double-blinded means that neither the patient nor the person administering or collecting data knows. Somebody else knows. You put all your information into a spreadsheet and then it gets sent off to somebody else who basically unencrypts it and figures out, oh, patient A137 was in fact in the placebo group. So we're going to sort that data out into the placebo group, where patient A329 was in the experimental group. So we'll put that data there. But again, that person didn't collect any data. They're just sorting it out and then the analysis is done. And it really does help remove bias. So now let's turn back to our experiment. This chart was actually published as part of their experimental design. Notice they started with 94 patients and a whole bunch of patients were excluded. In fact, they only had 24. And of those 24, they randomly sorted them in what's called a 1-to-1 ratio into the experimental group or the control group. So the experimental group receives ivermectin, the control group receives the placebo, 12 and 12. How many replicates do we have? We have 12. So this is 12 replicate. That's not very many. Notice all the reasons why people were excluded. They didn't want to participate. They were asymptomatic, so that was considered criterion for exclusion. They didn't live in the area. They didn't fit the age group. So here you can see the age group was between 18 and 60. 5 or more with 48 hours of fever or cough, too late in the course of illness. Severe disease, pneumonia is severe disease. Somebody who was already positive and that could conflict with the treatment plan because why were they positive 14 days ago and now they're coming back? And then somebody who looked like they had it but a PCR test showed negative, they had no baseline for that particular person. So you can see all these people that were excluded because of additional variables that the researchers wanted to control for. So what were the actual results? I encourage you to pause this and actually look at this for a little while. This shows you gene E and gene N are just two genes that were amplified. And they used the amplification of these two genes from the virus to basically look at viral load. Now notice this is a logarithmic scale, so every time you go up one, you're going up a factor of 10. It's actually a pretty big increase. The orange group received the true ivermectin. The blue group received the placebo. From day 1 to day 21, and you can see the averages. Now, one of the things about these types of graphs is that when you have those bars, in order to have what's called a significant difference, the bars need to be separate. They can't have overlap like that. So because they have overlap, they're showing that the results do not significantly support the hypothesis. In other words, ivermectin, a single dose of ivermectin, did not significantly reduce viral loads over those groups that got the placebo. And that was true regardless of which gene they picked. And they just picked two genes to kind of make sure that their data aligned. So how do you actually report these conclusions? So when you get your data, here are some different ways that you can report conclusions. Your results could just reject the hypothesis. Like it's clear that the hypothesis probably is not true. The results reject it. No follow-up is needed. There's no even indication of support. It's just like, no difference. And if we had seen like exactly the same viral loads, you could say the results reject the hypothesis. I think even with the data that you saw, you could say the results reject the hypothesis. Again, there's nothing wrong with being wrong. You cannot predict the future. That's why you did the experiment in the first place. If the bars had been actually separate and ivermectin was much lower than the placebo, then the results would support the hypothesis. You could say, yeah, a single dose of ivermectin significantly lowered the viral load. Again, remember we only had 12 replicates, so it's possible that if we had 300 replicates, you might have seen more of a difference, but 12 is a small number. Very often, you'll see something like the last bullet point. Results do not significantly support the hypothesis, meaning we don't have a statistical test to show that the difference is substantial, but some indicators suggest that there might actually be something there. So in other words, just because your data didn't support the hypothesis doesn't mean the hypothesis isn't true or can't be modified to be true with a different experiment. One of the other things that this particular experiment did is that they actually looked at self-reported symptoms. They looked at cough and basically changes in sleep patterns. And what you might notice here is that the top row is ivermectin, the bottom row is the placebo. And you'll see that later on, there seems to be less cough and better sleeping. So while the hypothesis was not supported, there was some indication that maybe ivermectin helped people feel better, at least later on in their course of illness. So with that, you could then revisit this, you could design a new experiment, you could try to expand it to include more people. In the news media, if you look at ivermectin, people will say, like, it's not worth it, it's not worth treating. And I would say that the data supports that general conclusion because you would want to see a significant difference in viral load, not that I felt better at day 20. So you would want to see a significant viral load to really promote that as a treatment. In other words, while it might help a little bit, it's not helping in a significant or substantial way, at least not based on the results that we just saw. So, other considerations: understand that science is constantly evolving. As we've moved through this pandemic, we're getting new information, and that in general, your physicians, the Center for Disease Control, World Health Organization, they're not out to get you. They have no profit motive in basically harming you. They're simply taking the best available science and putting it out there. And that has led, unfortunately, to kind of this idea that, you can't believe anything because it's always changing. Well, that's the very nature of science. You don't have all the answers right now. You get information and you make conclusions from them. And then you get more information, and sometimes old conclusions are thrown out and new conclusions are brought in. So that's the nature of science. If you really want to know, read the scientific papers, and they will show you how they did the experiments and what the data are, rather than waiting for somebody to condense it into your favorite news source. That probably has some sort of slant to it. Okay, one last thing, and that is just because the data does not support your hypothesis does not mean that you did something wrong and that you're going to fail or that you failed somehow. And it does also not mean human error. I read a number of student reports where they say, oh, the data did not support my hypothesis, human error. What human error? Did you do something wrong? Do you know that you did something wrong? No, but the data didn't support my hypothesis, so therefore your hypothesis was not supported by the data. That's the conclusion. Doesn't mean that your hypothesis is even false. It just means the data didn't support the hypothesis, and there's nothing wrong with that. But trying to cherry pick to basically build a house of cards around some idea is not good science. So don't do that. Rely on the data. If you don't trust the data, you better have a reason not to trust the data. That's more than just, it didn't give me the result I wanted. Okay, my next lecture is going to look at experimental design with masks. And this is one that I actually did. So you're going to have an opportunity to basically apply some of the concepts learned in this very long lecture on a real experiment that I did on the efficacy of masks. So I'll see you at the next one.